Hello everyone. It is my pleasure to present our recent work, titled Ribosomal RNA Operon Copy Number, a trait-informed framework to close the microbial cultivation gap, published in iMetaOmics. Our study establishes a correlation between ribosomal RNA operon, rRN, copy number and the culturability of microbial communities across diverse habitats. Conventional cultivation methods consistently favor taxa with high rRN copy numbers, leaving oligotrophic microbes with low copy numbers largely uncultured. Notably, rhizosphere communities host a higher high-rrn taxa, while gut microbiomes are enriched with medium to low-rrn organisms. These findings suggest that rRN copy number-based strategies hold promise for improving the isolation of functionally important yet difficult-to-culture microorganisms. Amplicon sequencing revealed that the dominant bacterial phyla in the rhizosphere, Proteobacteria, Firmicutes, and Actinobacteria, display crop-specific patterns. The soybean rhizosphere is predominantly composed of Proteobacteria and Firmicutes, whereas Actinobacteria show a higher relative abundance in maize. By further classifying bacterial communities into 6 categories, we found that the maize rhizosphere harbors greater proportions of permanently rare and intermediate taxa, while the soybean rhizosphere is richer in conditionally abundant taxa. Quantitative analysis revealed a strong positive correlation between rRN copy number and bacterial culturability. R superscript 2 equals 0.97 in soybean, R superscript 2 equals 0.99 in maize, matching the success rate of isolating amplicon sequence variants, ASVs, from both crops. Accordingly, Proteobacteria, typically high in copy number, were highly culturable, Firmicutes, very high copy number, dominated the permanently abundant group, and Actinobacteria, moderate copy number, exhibited genus-level variation in culturability. We also demonstrated that sample preservation conditions significantly affect microbial recovery. Fresh samples captured the broadest diversity. Refrigeration without cryoprotectants reduced Proteobacteria recovery and enriched Actinobacteria, while glycerol partially restored Proteobacteria isolation. Importantly, low-rrn taxa were consistently depleted across all option treatments, highlighting a compounded bias from both cultivation and storage. Comparing habitats, rhizosphere communities had a higher average rRN copy number, 6 to 8, than gut communities, 3 to 5. The rhizosphere also contained more high rRN and fewer low rRN taxa than the gut. Furthermore, the culturability of different rarity categories correlated positively with their mean rRN copy number. Culturable taxa consistently possessed higher average rRN copy numbers than unculturable ones. Microbial doubling time was negatively correlated with rRN copy number, indicating that high rRN taxa are not only more culturable but also grow faster. Based on these traits, we propose several targeted cultivation adjustments, using oligotrophic or chemically defined media to favor low-rrn and slow-growing taxa, extending incubation times, simulating natural nutrient fluctuations, tailoring temperature and oxygen conditions, employing diffusion chambers or iChip devices to maintain microbial interactions, applying helper strains for cross-feeding or detoxification, and optimizing cryopreservation protocols to minimize the loss of low rRN taxa. In summary, the well-known plate count anomaly and inherent biases in traditional methods have left many oligotrophic microbes uncultured, limiting both applications and biodiversity conservation. Our work shows that rRN copy number is a strong predictor of this cultivation bias and that trait-informed strategies can enhance the isolation of both abundant and recalcitrant microbes. While a powerful rRN copy number should be integrated with other genomic traits, e.g. genome size, for a more comprehensive cultivation framework, this trait-based approach effectively bridges sequencing data with cultivation practice, offering a pathway to access the hidden microbial majority. Future studies should focus on elucidating the causal mechanisms linking rRN copy number to microbial culturability,